Good evening. So I welcome you to the week 11 live session of experimental biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working at the Department of Biotechnology in IIT Kharagpur. In today's session, we are going to talk about protein-ligand binding, fluorescence anisotropy, electrophoretic mobility shift assay, isothermal titration calorimetry, and surface plasmon resonance. So these techniques we are going to discuss in light of the protein-ligand binding and how to determine the dissociation constant in this case of when a protein binds to a ligand, and what are the thermodynamic parameters that we can calculate when a protein binds to a ligand. So we are going to discuss that in today's session. Okay, so when a protein binds to a ligand molecule, so usually you can understand that the ligand molecule is much smaller in size than the protein molecule. And so the ligand, when it binds to a particular binding site of the protein, and this reaction is reversible reaction. So the ligand binds to the protein. This reaction is the association reaction, and the rate constant of this reaction is denoted by K on. So K on is the association constant, the rate constant for the reaction of association. Whereas when this ligand can be dissociated from the protein, so the reverse reaction, it has a rate constant of K off. So K on and K off. These rate constants are written in small letters. Remember that. So now we can see that when a protein and a ligand, they associate with each other, they form a protein-ligand complex. So the protein and the ligand, when they associate with each other, they form this protein-ligand complex. And this is free protein, this is free ligand. So, and this is the bound complex between the protein and the ligand. Now, the rate of change of the concentration of this protein-ligand complex, that is dPL by dt, which is the rate of change of concentration of the protein-ligand complex. This can be written as K on into P into L. So this is the forward reaction where the protein and the ligand bind to each other, minus the reverse reaction, which is the K off into PL. So when the protein ligand complex dissociates to form free protein and free ligand, right? So this is the equation that we can get when a protein binds to a ligand. And remember that at equilibrium, this rate of change of protein ligand complex becomes 0. So at equilibrium, this factor becomes 0. So there is no rate of change of there is no change in the protein ligand concentration when the association and the dissociation rates become same. Okay, so then what happens? Then we can write K on into P into L equals to K off into PL, right? And therefore, what we can write is that K off by K on, we can write as PL by, that is the protein ligand complex concentration divided by the concentration of the free protein into the concentration of the free ligand. So this is how we can represent K off by K on. And we know this term stands for the dissociation constant, which is Kd, right? So therefore, we can write this that K off by K on will be equal to Kd at equilibrium. Now, how do we determine the dissociation constant? What is the experiment that we can design? So the experiment that we can design here is a titration experiment. So suppose in a conical like that, we keep the, so this is my protein solution, and above this I can have a syringe, or I can have a bullet, or I can have some device where I have the ligand. Now the ligand is titrated drop by drop into the protein solution, right? So the ligand is titrated into the protein solution. Now this is the typical experimental setup. The reverse can also be done when the ligand is in the solution and the protein can be titrated. So it depends on which factor we want to observe on the experiment. So here, if we have a protein solution and we are drop by drop increasing the or drop by drop titrating the ligand into the protein solution, then what we get? So in the beginning, we know the total ligand concentration written as LT. So total ligand concentration is known to us, but we do not know. So this will be the stock solution of the ligand. But when this ligand is added drop by drop into the protein solution, we do not know what is the concentration of that particular ligand, right, in the protein solution. So that we do not have any information, but we have information of the total protein concentration, right? So total protein concentration is known to us. Here, so what we do is we add the ligand drop by drop, and when we keep adding on the ligand, so what happens, initially there is no ligand, so the proteins are all unbound. But then when ligand keeps on coming in the solution, then the protein molecules, they start to bind to the ligand. And so if we plot it in a graph, so in this kind of a graph, if we plot it, right? So here in the x-axis, we have total ligand concentration, and here in the y-axis, we have the concentration of the protein ligand complex. So what we will see at the time t equals to 0, there is no ligand, so there is no protein ligand complex, so it is also 0. But as we keep on adding the ligand, so then what happens? So suppose we add this much of ligand, then protein ligand complex starts to form, and as we keep on increasing the ligand amount, so then what will happen? The protein ligand concentration will also increase. The complex concentration will also increase, but at a certain point, it will not increase any further. So here we can see this protein ligand complex formation is being saturated. So why is this being saturated? Because in the protein solution, the amount of protein is fixed. So as you are increasing the ligand concentration, slowly what will happen? All the proteins will be occupied with the ligands. So there will be no more proteins left to bind to the ligand. So even then, when you increase the ligand concentration, no more proteins will bind to the ligand. So therefore, the protein ligand complex, this will remain constant. So that is when the saturation is reached. So if we plot this kind of a graph where we plot the protein ligand complex concentration with respect to the total ligand concentration, we can see this kind of a graph where it will reach saturation after some time. And from this particular experiment, so these are the experimental points, right? So these are the experimental points. So this is the ligand concentration that you are adding. So from this experiment, how can we calculate the dissociation constant, that is the Kd? So in here we mentioned that Kd is what? Kd is the protein ligand complex concentration divided by the free protein concentration into the free ligand concentration, right? So that is Kd. So now we have information about the total ligand concentration and total protein concentration, right? But we do not have any information about the free protein concentration as well as the free ligand concentration. So now how can we get that? So we know that total protein concentration is what? Total protein concentration is free protein plus protein ligand complex, right? So this is the summation of free protein plus protein ligand complex. Again, the total ligand concentration is also the summation of free ligand plus protein ligand complex, right? So these now we can put in this particular equation, right? So in case of this free protein concentration, we can write free protein concentration equal to protein total concentration minus protein ligand concentration, this, right? And in case of free ligand concentration, what we can write is from this particular equation, free ligand concentration is ligand total minus protein ligand concentration. And these two we can put in this particular equation, and then that will give us the value of the dissociation constant, or Kd. So if we solve this equation, so this is a quadratic equation, and if we solve the equation, we will get this kind of a equation. So which will give us the protein ligand concentration, protein ligand complex concentration. And here in this particular formula, what you can see here is that this protein total concentration is known, ligand total concentration is known, and Kd value is not known. So protein ligand concentration, we will have an idea of the protein ligand complex concentration. Why? Because the experiment that we design it such that the protein ligand complex concentration can be detected. So therefore, if we have this idea of the protein ligand complex concentration, from that we can get the value of the dissociation constant by solving this particular formula. So by this way, we can calculate the dissociation constant of the protein ligand binding by a titration experiment. Now, there are some considerations that we have to always take while we design an experiment for this calculation of Kd. What is this consideration? That is that we should have an idea about the range of Kd that may result in our experiment. So we know our protein, we know our ligand, and we should have a preliminary idea of the range in which this protein and ligand will interact. So their binding, we should have a idea about the Kd, or dissociation constant, range. So which means that whether it is in nanomolar range, or whether it is in micromolar range, or whether it is in millimolar range, we should have an idea about it so that we can design our experiments in such a way. So why is it necessary? So first of all, before going to that, let me just discuss the unit of Kd. So Kd is, you can see that Kd is concentration of protein by concentration of ligand divided by the protein ligand concentration. So therefore, Kd has an unit of concentration. So concentration into concentration divided by concentration. So of course, this means that the unit of Kd will be that of concentration. So it can be nanomolar, it can be micromolar, or it can be picomolar. Now, the binding of these molecules, that is, a protein can bind to a small molecule, a protein can bind to another protein, a protein can bind to a nucleic acid. So these kind of bindings, so there is difference in the strength of these bindings. So this binding can be either very tight, it can be tight, it can be moderate or weak binding. And depending on that, obviously our Kd will vary. So lower Kd value means tighter binding. So therefore, when the binding of the protein and the ligand is very tight, then the Kd will be in the nanomolar range. So it will be less than 10 nanomolar. So in case of tight binding, it can range from, the Kd value can range from nanomolar to 1 micromolar. In case of moderate binding, the Kd value will increase further. So it will be around 1 micromolar to 100 micromolar. Whereas in case of weak binding, the Kd value is much greater than that. So it's approximately 100 micromolar or so. It can be greater than 100 micromolar. So now we can guess that since the Kd value has a broad range, so it can be from nanomolar to micromolar range. So therefore, the sensitivity of our experiment also has to be such that it can detect this range of Kd. So in case of very tight binding, the sensitivity must be in the range of picomolar. So how do we determine that? So if we are designing an experiment and the protein ligand complex Kd is more or less, we can assume a Kd. So then the range in which the experiment should be done, or the range of sensitivity of that particular instrument or the experiment should be in this range 0.1 Kd to 100 Kd. So suppose our protein ligand complex has a dissociation constant of 1 nanomolar. So if the dissociation constant is 1 nanomolar, then the range of sensitivity of that experiment should range from 0.1 Kd, which means 0.1 nanomolar, to 100 Kd, which means 100 nanomolar. Right? So this is the range that should be taken into consideration while designing the experiment. So of course, you can assume therefore that for very tight binding, the range should be in picomolar. For tight binding, the range should be in nanomolar. The sensitivity should be nanomolar. For moderate and weak binding, the sensitivity should be in micromolar or millimolar range. So these are the considerations that should be taken care of before designing an experiment to determine the value of Kd of a protein ligand binding. Now we will one by one discuss the experiments which are which we can use for uh, determining the Kd of a protein and ligand binding. So the first experiment is fluorescence anisotropy. So what we do in case of fluorescence anisotropy is that the basic principle of fluorescence anisotropy is that there is a polarized light. So the polarized light is passed through the ligand solution. So here you have to remember that here the ligand is labeled. So mostly it is fluorescently labeled. So this is the ligand. The ligand is having a fluorescent label on it. And when we pass polarized light through this ligand. So this ligand is a small molecule. So if this is a small molecule, then it will have rotational diffusion in the solution, right? So it will rotate very fast in the solution, and therefore the ligand molecule, because of this rotational diffusion, when we pass this polarized light through it, this ligand will be excited, but when it will be emitting the light, it will emit a depolarized emission. Why? Because of this rotational diffusion of the ligand molecules. So they will rotate in different directions, and of the light that will be emitted from them will be a depolarized emission. Now, this is the situation when there is only ligand and there is no protein. Now, next thing what we will do is we will titrate the protein into the solution. Okay, so we will keep on increasing the protein concentration, and when the protein will come into the solution, the protein will bind to the fluorescently labeled ligand. Now, because the protein is a much bigger complex than the ligand molecule, so once the protein binds the ligand, there will be very slow molecular rotation. So the rotational diffusion will be less, because of which the depolarized emission will be much less in case of a protein DNA or the protein ligand complex. So therefore, you can understand that when the protein binds to the ligand, the the emission will be still polarized. So therefore, the difference between this fluorescence polarization can be um, can be actually measured. So this depolarized emission minus this polarized emission, so the difference between this can be measured, and then from this measurement we can have an idea of the KD value. So how do we do that? So this fluorescence anisotropy experiment. So here this in the y-axis, we plot the difference between the polarization of the protein ligand complex and the free ligand, right? And this we plot with respect to the total protein concentration. So why do we plot with respect to the total protein concentration? Because here we are titrating the protein, right? And if we plot this kind of graph, we will see. So these are the experimental points. We will see a very similar graph to that of protein ligand binding graph. So initially, the with increase in protein ligand concentration, this difference will increase, and then it will slowly saturate. So from this graph, again, using the same equation, we can calculate the dissociation constant. And this fluorescence anisotropy experiment, it works well in the moderate affinity range, which is in the affinity of, which has the affinity of 0.1 micromolar to 100 micromolar. And here, by putting it into this equation, we can calculate the dissociation constant. And this is the response or the absorbance values that we get. And we use this for normalizing this um, equation. And from this equation, we can calculate the value of Kd. So any doubts so far? Okay. Now we will move on to the next experiment, which is Electrophoretic Mobility Shift Assay, or EMSA. So EMSA is performed to study protein nucleic acid interaction. In this case of fluorescence anisotropy, this could have been interaction between protein DNA, or this could have been interaction between protein small molecule. Many types of interactions can be observed and determined by this fluorescence anisotropy, whereas electrophoretic mobility shift assay is an assay to study the protein nucleic acid interaction. So here, what we do is that we have these series of tubes here. And in these tubes, we have the same concentration of DNA. Here, this DNA is labeled. What does it mean? So this DNA has a particular label. It can be a radioactive isotope, or it can be a fluorescent label as well. So a radioactive isotope means gamma 32P ATP. Gamma 32P ATP. It is a radio-labeled isotope. It is a radioactive isotope that is used to label DNA. And the radio-labeled isotopes have actually very high sensitivity, up to 10 to the power minus 18 molar, which is why the radioisotope is actually used to label DNA. But remember that when we label DNA with radioisotope, since it emits radioactive emissions, so of course we have to be very careful with while handling this kind of radioisotopes. And this experiment will also need some expertise and some standardization. Now, in these tubes, we have the same amount and the same concentration of this labeled DNA. And in these tubes, we sequentially add protein in increasing concentration. So here we keep the volume of this sample constant, but we keep on increasing the protein concentration, right? And then these samples, these samples will be run in native page. So native page is the non-denaturing page where there will be no use of HDS, right? So therefore, the page is not denaturing in nature, right? So these samples are now run in the non-denaturing page or native page, and what we observe here is this kind of an figure, this kind of an image from the gel. So here, this lane A denotes the free DNA. So this is the free DNA. That means there was no protein which was added here. And from in this particular direction, the protein concentration is increasing. So the sorry. The protein concentration is increasing in this particular direction, right? So what we observe here is that as we increase the protein concentration, what we observe is that this free DNA keeps on, the intensity of the free DNA keeps on decreasing, right? So what we observe here is that the intensity of these bands keeps on decreasing in this direction, right? Whereas this particular band, the upper band, this indicates the protein DNA complex. This indicates the protein DNA complex, and as we can observe here is that when we keep on increasing in this direction, when we keep on increasing the protein concentration, more and more proteins are binding to the DNA, and therefore the free DNA concentration is decreasing while the protein DNA complex concentration is increasing. So therefore we can see an increase in the intensity of the bands of the protein DNA complex and the decrease in the intensity of the free DNA band. Next, so by which we can understand that the particular protein is binding to the DNA molecule. Now, therefore, how will we determine the dissociation constant from this kind of an experiment. So IMSA is a very sensitive experiment, and it can actually determine the dissociation constant uh, in a very dissociation constant of very low range, right? That means for very tight binding. So here, how do we determine the dissociation constant? So here also we will get a similar kind of graph when we plot the saturation, and this is the same equation that we will use here, and from that we can get the value of the dissociation constant by plotting the same kind of equation. And the beauty of IMSA is that uh, small to large, very small to very large protein nucleic acid complexes can be studied by this kind of an assay. That means from 20 kilodalton to 1000 kilodalton large complex can also be run in the gel. And it's a very highly sensitive experiment. And there might occur a little subtle problem in this IMSA, that is, when when these samples are run on the gel, so then due to the electrophoresis, now during electrophoresis, this protein DNA complex may dissociate, and therefore this particular bands, the protein DNA complex bands, these bands may decrease, or these bands may appear blotchy because of the dissociation of the protein and DNA. So therefore, um, it is essential to keep in mind that here in this particular experiment, even though we can see the free ligand and the bound ligand, the concentration of the free ligand as well as the concentration of bound ligand simultaneously we can detect, but detection of the concentration of bound ligand of the protein DNA complex is dependent on the appearance of the on the gel, right? So therefore, what we do here is we estimate, or we actually calculate the intensity of the free DNA band, because this does not change with electrophoresis, right? So the free DNA band intensity is actually measured, and this free DNA band intensity is plotted, right? So there this problem might arise in IMSA, so therefore it requires some standardization, and this requires repeated experiment so that the bands appear crisp. Okay, so now we will move on to the next experiment, which is isothermal titration calorimetry, or ITC. So isothermal titration calorimetry is an experiment where we can detect the Kd value, that is the dissociation constant, and the thermodynamic parameters like the enthalpy, the change in enthalpy, change in entropy, change in free energy simultaneously from one experiment. So let's look at the basics of this particular experiment. So here we have an isothermal chamber. So in this isothermal chamber, there are two cells. One is a reference cell, another is a sample cell. So in the reference cell, we have our protein, and this protein is immersed in a buffer solution, right? So in the reference cell, we have our protein in buffer. We have our protein in the buffer, and in the sample cell, we have our protein where the ligand is titrated. So here on top of this, you can see a syringe. In this syringe, we take our ligand, and this syringe is adding the ligand slowly into this sample cell, right? And there is a needle which is present in here to mix the ligand with the protein, right? And this is the isothermal chamber, which means the temperature of the chamber is kept constant by the help of this adiabatic shield. Now, in this experiment, what happens is that when we titrate the ligand in the protein solution, we get uh, we get a measurement of the heat evolved or the heat absorbed. So the heat change uh, can be calculated. So how do we see the response from this kind of an experiment? So this is the raw data that we can see. So it kind of looks like this. Or it may be in the 1 minute. So actually, when we are adding the ligand in the protein solution, there is a heat change, and this heat change can be calculated by this area under the curve. So we can see this kind of peaks, peak 1, 2, 3, 4, and by calculating the area under these peaks, we can calculate the heat change. That is the heat evolved or the heat which is absorbed. So this kind of from this this is the raw data of this experiment where we can get the measurement of the heat change, right? So this heat change can be measured here. Now this this heat change that is in the kilojoule per mole or kilocalorie per mole, it can be plotted in this kind of a graph, right? With respect to the molar ratio. So with respect to the molar ratio, we can plot this graph, and what we observe here is this graph again looks like this, very similar to the protein ligand binding graph. And from this graph, we can determine both Kd as well as delta G and delta H. So Kd can be determined by putting it in the same equation. And when we determine Kd, we can also have an idea of the delta G, because delta G will be equals to minus RT ln K. So from that, we can get an idea of delta G. And if we have the idea of delta G and this heat change, the change in this heat, so this is the heat in the y-axis, and this change in the heat will give us the delta H. So the change in enthalpy, the delta H, we can get the delta H from this particular graph. And when we have both delta G and delta H, we can also calculate the change in entropy, that is delta S. So all the thermodynamic parameters, that is the delta G, delta H, and delta S, as well as the dissociation constant, can be determined from this kind of experiment, isothermal titration calorimetry. Any doubts so far? Okay. So now we will move on to another part of our discussion, that is how do we determine the kinetics, which is the rates of reaction, the K on and K off. So before going to that, first just let me discuss once again about this particular equation which we were dis discussing in the very beginning, that is the change in the protein ligand complex concentration will be equal to the K on into protein and ligand concentration, the multiplication, the product of the protein and free ligand concentration, minus K of minus the reverse reaction, which is the K of into the protein ligand complex concentration. Right. So by dimensional analysis, we can calculate the units of K on and K off. So what is the unit of the change of protein ligand complex concentration? That should be concentration over time, right? So that is the uh, rate of change of the protein ligand complex concentration. So that it has a unit of concentration over time. So this part will also have a unit of concentration over time. So therefore, here the unit of K on will be 1 by time into concentration, whereas the unit of K off will be 1 by time. So this K on and K off, they have two different units. Now, how to determine this K off? So actually, if we have to determine the rate of this reverse reaction, then we have to make sure that the reverse reaction is only being carried out and this reverse reaction becomes irreversible. This becomes irreversible, right? So we have to create a design an experiment in such a way that this reverse reaction is only occurring, which means the protein ligand complex is uh, dissociating to give the free protein and the free ligand. So we have to design an experiment in this way so that this becomes irreversible. Now, when this becomes irreversible, so what we can write, the change in the ligand concentration will be equal to K off into protein ligand complex concentration, right? And in place of this protein ligand complex concentration, what we can write, PL is equal to ligand total minus free ligand, right? So this we can substitute here, and when we integrate this equation, we get this kind of an when we integrate this equation, we get this formula, which is L equals to the free ligand concentration equals to ligand total concentration into 1 minus e to the power minus K of into T. So this becomes an exponential um, equation where, and if we plot it, so if we now titrate the ligand, so with respect to time, what we will see is that the free ligand concentration will actually saturate with time, right? So from this experiment, we can determine the K off or the dissociation rate. So for determining this dissociation rate, one thing that we must remember is that we have to carry out the experiment in such a way that we can accurately determine the dissociation rate. For that, there are two parameters that we have to consider. One is how long we should run the experiment, and another is how fast we should collect the data. So the K off, I told you that this K off has this unit of per second, right? So it has this unit of 1 by time. So that means this K off has a unit of per second or per millisecond or something like that. But this concept of per second is not very intuitive. So we must have an idea of the time for which for 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 how much time we should carry out the experiment. For that, what we do is we take another constant, which is the time constant, which is tau, which is nothing but 1 by k of. So it is the reciprocal of k of. Now k of equals to, suppose if k of equals to 0.2 per second, then tau will be 1 by 0.2, which is tau will be 5 seconds, right? So why do we calculate this time constant? So that we can have an idea of how long we have to run the experiment. So if the tau value is 5 seconds, then we should at least collect data up to 4 tau value, right? So up to 4 tau value, we should collect the data, which means if the tau is 5 seconds, we should at least collect the data up to 20 seconds. So we can see it here in this particular graph that suppose this has a tau value of 5 seconds. So after 5 seconds, we can see that after 5 seconds, only 63.2% of the reaction is completed, right? Now, when we go to 2 tau, that means 10 seconds, right? 2 into 5, that means 10 seconds, 86.5% of the reaction is completed. When we go to 3 tau values, we see that 95% of the reaction is completed. Whereas when we go to 4 tau value, up to 4 tau value, we can see that approximately 98% of the reaction is completed. So which is why we should always collect data up to at least 4 tau value. Otherwise, the tau value, otherwise the um, observation will be erroneous. So there will be errors. So at least we should collect the data for up to 4 tau value. Now, the next question which comes is how fast we should collect our data. So of course, if it depends on the binding kinetics. So if the binding is fast, then we should collect data faster. If the binding is slow, then we can collect data slower, right? We do not need to collect data that much fast, but it has to be remembered that in case of slow binding, it takes long time for the reaction to be completed or to reach saturation. So for fast binding, we should collect data faster, and the rate of data collection should be 2 tau by 5, right? So if our tau value is 5 seconds, then the rate of data collection should be 2 into 5 by 5, which means approximately 2 seconds. So in the interval of 2 seconds, we should collect the data. That will give us a better estimate of the k off rate, right? And after collecting this data, we can plot it in the same equation. And in this particular equation, we can plot the plot the values, or we can put the values in this equation, and then we can calculate the k off, or the rate of dissociation of this complex. Now we are going to discuss the experiments by which we can calculate this k off value. So the first experiment we are going to discuss is EMSA. EMSA I have already discussed, but here for determination of the k off, there is a little bit difference in than that of the previous EMSA experiment that I discussed. Here we have to remember that this reverse reaction has to be dominant. This reverse reaction has to be dominant. So for maintaining that, what we do here is that we take the labeled DNA and then take our protein, and also we add approximately 100 folds excess of unlabeled DNA. So this is unlabeled. This is unlabeled DNA, and this is labeled DNA. So why do we take unlabeled DNA? So which means that when the protein, when the protein is added in this reaction, then the protein will bind to the labeled DNA. But once the protein dissociates from the labeled DNA, it will, the chance of it to bind to labeled DNA will be much, much less because the unlabeled DNA will be in much excess. And this unlabeled DNA is used so that this reaction can be maintained irreversible, right? So that this dissociation, when this protein dissociates from the labeled DNA, this dissociation becomes almost irreversible because the chance of the protein to bind to labeled DNA again is very, very less because the unlabeled DNA is much more in excess than the labeled DNA. Now, this labeled DNA, the intensity of the labeled DNA we will detect by gel electrophoresis by native page. So therefore, this labeled DNA will be free, and so this will be our free ligand, right? And this experiment uh, we will actually carry out for some time, and while this experiment, while this reaction is going on, we will do, what we will do is that at different time points we will take 15-20 microliter of the sample from this reaction, and then we will keep on running it in the gel. So while this native page, so this is again native page, while this gel is running, in between that, we will keep on adding the some amount of the sample at particular time interval. So suppose we add it at t equals to 0. In the first lane, we add the sample at t equals to 0. And in the next lane, we will add it after, suppose, 2 minutes. So then T1 will be 2 minutes, then T2 will be 4 minutes. So at 2 minutes interval, we are adding the sample on the gel while the gel is running. So this has to have a considerable amount of expertise because the gel will has to be the samples has to be done in the gel while the gel is running. And why is it done? Because with time, what will happen? The more and more labeled DNA will be freed because this reaction becomes almost irreversible, right? So more and more DNA will be freed up, right? And as we keep on adding the samples after a particular time interval, we will see that the free DNA concentration will increase. We can see here that this free DNA concentration will increase when we go from left side to the right side. So as we proceed in this reaction, the free DNA concentration will increase. Why is it increasing? Because the labeled DNA, which is dissociated from the protein, it cannot bind to the protein again because there is much more unlabeled DNA in this reaction, right? And since this free DNA, uh, so what happens here is that this lane is run first. This particular lane is run first, and then followed by this lane 2, lane 3, lane 4, lane 5 at particular time intervals. So of course, the last lane will have the least migration because it has run for least time. So here the migration will be less, and here the migration will be more. And from this particular gel, what we can calculate is that we can calculate the free ligand concentration. So the free DNA concentration can be calculated, right? And this intensity of the free DNA concentration can be plotted with respect to time, right? So we can see that with increase in time, we can see that the free ligand concentration is increasing, right? And after a certain point, it is becoming saturated. So when there is no more free DNA, right, or no more proteins to bind to the DNA, right? So this happens, and from this kind of plot, we can again calculate the K-off rate from by using the same equation, right? So that is how we can determine K-off by M sub. And here I would just like to mention one thing, that to determine K-off by M sub, the experiment. So here we can understand that we are adding the samples at different time points, and these time points will be in minutes, not in seconds or milliseconds, right? So for the K-off, if we have to determine the K-off by IMSA, then that means the reaction has to be slow enough so that so it has to have a slow binding kinetics, so that we can if we keep on adding the sample at 2 minutes or 5 minutes interval, we can still calculate the K-off from it, right? So that is the limitation of this kind of an experiment. Now, the next experiment that I'm going to discuss does not have any such limitation of IMSA. So this particular experiment, which is the surface plasmon resonance SPR, it can be used to detect a broad range of kinetics from fast to slow binding. So what is the setup of this experiment? So we have a flow chamber. So this is the flow chamber. We have a flow chamber, and inside this flow chamber, we have immobilized our proteins. So our proteins of interest are immobilized inside the flow chamber. On top of the flow chamber, we have a thin layer of gold. So it's a metallic layer. And on top of that, we have a sensor chip. Now, so when we run buffer through this flow chamber, what happens that the proteins do not interact with buffer, do not bind to the buffer. So the buffer flows through this chamber, and when the buffer flows through this chamber, there is no change in the refractive index. There is no change in the refractive index. So what happens is that we pass a polarized light. So the polarized light, it hits on this gold film, and then it is totally internally reflected. So now we can calculate the angle for this total internal reflection, right? Now, when there is buffer flowing through this flow chamber, there is no deflection in the reflected light, right? But when we pass ligands through this flow chamber, so after buffer, what we do is we pass ligands through this flow chamber. So now these ligands, the proteins will bind to these ligands, and once the proteins bind to the ligands, then what will happen? Because of this protein ligand binding, there will be a response generated. So because of this protein ligand binding response, there will be a change in the angle for this total internal reflection to occur, and due to this change in the refractive index of the medium. So the refractive index of the medium will change because of the binding of the protein with the ligand, which is why there will be a change in angle of the total internal reflection. Now, this change of angle can be detected by a detector, and this can be plotted. Now, the resonance signal or the response of this kind of an experiment looks like this. This is how it looks like. So here what happens when the buffer is running through the flow chamber, we can see a steady response, which is the baseline, right? Now, when we pass ligand through this flow chamber, then we can see an association. So, which means that the resonance signal keeps on increasing, and after a certain point, it reaches saturation. So, therefore, it means that this sample, when this ligand is passing through the flow chamber, the ligand is binding to the protein, and due to this protein ligand binding, this response is actually increasing. And after a certain point, when there is no more ligand, no more, when there is no more protein available for binding to the ligand, then this signal flattens or saturates. Now, after this point, when this signal flattens or saturates, then what we do is we again pass the buffer through the flow chamber. So now the buffer is again passing through the flow chamber, and as buffer is passed, which means that again the dissociation reaction is becoming irreversible. So when the buffer is passed, the ligands which are dissociated from the protein, these ligands will flow through and flow out of the flow chamber because of the running of the buffer, right? So the dissociation now becomes irreversible. The ligands which are dissociating from the protein, they cannot bind to proteins anymore. So the reaction becomes irreversible, and then this decrease in the response we can observe. This decrease in the response we can observe when this dissociation becomes irreversible. So we can observe a decrease in the response, right? So this is called the dissociation curve. So this part is the association curve, and this is the dissociation curve, and then this signal comes to the baseline, right? So this is how we can get the response from an SPR experiment, the response or the resonance signal from the SPR experiment. Now, how do we determine the thermodynamic and kinetic parameters from this SPR experiment? So what we do here is that we keep on increasing the ligand concentration, right? So as we go up, we can see that the ligand concentration is increasing. And once the ligand concentration is increasing, so therefore what happens, the more and more ligands are flowing through the flow chamber. So therefore, the response will be also higher because more and more proteins will bind to the ligand. So we can see that when we increase the ligand concentration, the response also becomes higher, right? And this response is plotted against the time, right? And this maximum response units, the maximum value of this response now can be plotted with respect to the ligand concentration, the total ligand concentration which is known to us. And this response unit, when it is plotted against the ligand concentration, this will give rise to this kind of a graph, which is very familiar to us. From this graph, we can calculate the KD value, right? And this particular experiment, the SPR, can be used for a broad range of dissociation constant from 1 nanomolar to 10 micromolar, right? And as well as fast kinetics can also be measured. So now, how can we determine the kinetic parameters? How can we determine the kinetic parameters of the experiment? So as I told you that when we are passing only buffer through the flow chamber, so then what will happen? So this dissociation will begin. So this dissociation occurs because there is the reaction becomes irreversible. So the ligands, when they are dissociating from the protein, they cannot bind to proteins anymore. So they are flowing out of the chamber, which is why we can see a decrease in the response signal, right? So therefore, this particular reaction becomes irreversible. So PL dissociates to give rise to free protein and free ligand. So from this, we can calculate the K of value, right? So del PL by dt, del PL by del T will be equals to minus K of into protein ligand concentration, from which we can calculate. After integrating this particular equation, we can calculate this K of value, and this is the response unit. So response unit will be equals to response unit max into e to the power minus K of into T, right? So from this equation, we will be able to calculate the K of value. So in this particular experiment, very fast kinetics can be measured as well as slow kinetics can be measured, and we get this idea of thermodynamic parameters as well as kinetic parameters from this experiment. Any doubts so far? All right. So now we will proceed with the question answers. So this is the first question. Please let me know your answer. Dissociation constant has the unit of concentration. So I mentioned this, that dissociation constant is, so therefore it has unit of concentration into concentration by concentration. Yes, so dissociation constant has the unit of concentration. Yes, A is the correct answer. Okay, coming to the next question. Yes, the correct answer is native polyacrylamide gel. Okay, next question. Yes. So for determining binding kinetics, actually we can use all of these experiments. INSA, ITC, anisotropy, surface plasmon resonance, all of these experiments can be done to determine the binding kinetics. Okay, next question. Yes, the answer is correct. ITC. Next question. So, free energy of a binding reaction can be calculated again from all of these experiments. Moving on to the next question. Yes, this is the next question. Yes, right answer. Next question. So, which is the parameter which is detected in fluorescence anisotropy? So, here the correct answer is B. That means the decrease in rotational diffusion of the small molecule is detected. Decrease in rotational diffusion because when the molecule when the molecule is free, it is rotating in all directions, but when the molecule is bound by a protein, its diffusion becomes limited, right? So the decrease in the diffusion of the small molecule is detected, and which of in these statements, the other correct statement is C, which is the fluorescent label is excited by polarized light, right? Which helps in detection of the decrease in rotational diffusion of the small molecule. B and C are the correct answers. Next question. So what happens in presence of competing inhibitor? When there is an inhibitor which competes with the ligand, then what will happen to the dissociation constant? Will it increase or will it decrease? Okay. The solution should be 10 to the power minus 5 molar. So if Ka is 10 to the power 6 mole inverse, then Kd will be 10 to the power minus 6 mole without any without any inhibitor. But when there is an inhibitor which is present, then what do you think? What will happen to the Kd value? So if the Kd value decreases, if the Kd value is low, which means the binding is tight. And if the Kd value is high, then it means a weak binding. So in without a competing inhibitor, the Kd value will be 10 to the power minus 6 molar, right? But when there is an inhibitor present, obviously the binding will be weaker when there is an inhibitor present, right? So if the binding is weaker, then the Kd value will increase. And here, the only Kd value which is higher than 10 to the power minus 6 is option A. So we can assume that Kd value of 10 to the power 5 molar is not correct. It is in the positive, so it will not be correct. 10 to the power minus 6 is when there is no inhibitor. 10 to the power minus 7 is when the binding becomes more tight. And this does not happen in presence of an inhibitor. So in presence of an inhibitor, the binding becomes weaker, so the Kd value will increase. So the correct solution will be A. Is that all right? Coming to the next question. What can we calculate from the Kd? Actually, the correct answer is A, B, and C. So from Kd, I told you that delta G equals to minus RTL and K. So from that, we can calculate delta G value from Kd. We can also calculate the Ka from Kd. And we can also calculate the concentration of ligand which is required for the half maximal occupancy of the protein. Okay, so all these three can be calculated from Kd. But K on and K off cannot be calculated directly from Kd. Next question. The correct answers are A, B, and D. So the ligand constitution can be varied, the ligand constitution can be kept constant, and the protein constitution can be varied. So both of these can be done, A and D. Okay, and also it is necessary to keep the temperature constant. So the correct answers are A, B, and D. So either the ligand can be titrated in or the protein can be titrated in. So if the ligand is titrated in, then the ligand concentration is varied. If the protein is titrated in, the protein concentration is varied. And it is also essential to remain the to make keep the temperature constant. Calculate the dissociation rate of dissociation constant. Next question. Yes, protein nucleic acid interaction. Correct. Next question. So how can we calculate tau? Tau is 1 by k of. Tau is 1 by k of. This 1 millisecond. Next question. What is the relationship between tau and k of? The correct answer is, of course, C. Data should be collected for at least 40 milliseconds or longer, and data should be collected at so every 4 milliseconds or faster. That is also a correct option, right? Because it is actually calculating for how long the data should be collected and for at which interval the data should be collected. Okay, next question. The correct answer here is no. The correct answer here is um, surface plasmon resonance. It's the best way to study the kinetics. The key of. The next question. A low dissociation constant means what kind of binding? Tight binding or weak binding? Yes, the correct answer is A binds P more tightly. Next question. So here the chaos and the kaons are given. Now, what can you conclude about this binding interaction? Here the correct answer is B and C. So, the thing is that A has an association constant of 10 to the power 4, where B has an association constant of 10 to the power 7. So B will bind to the protein, B will bind to the protein P more tightly because we know that K off by K on equals to KD, and K on has a limitation. K on has a limitation of the diffusion. So it is limited to 10 to the power 9 per molar per second. That is the maximum K on that can be achieved. It is the limit of diffusion rate. So therefore, if A has a dissociation constant, A has an association constant of 10 to the power 4 and dissociation rate is 0.001. That means 10 to the power minus 3, right? And divided by 10 to the power 4, which is for A. For B, which is 10 to the power minus 1 divided by 10 to the power 7. So from here, we can have an idea of the KD. So B will have a lesser KD. B will have a lesser KD. So B will bind to the protein more tightly. B will bind to the protein more tightly. Whereas the AP complex is longer lived than the PP complex because the dissociation rate of AP complex is much less than the dissociation rate of the BP complex, right? So which is why B and C will be the correct answers. Okay, and last question for today. This is the equation that we discussed. Yes, the correct answer is A. All right. So with this, we end this session for today, and we will have our last session in the coming Tuesday for week 12. And thank you so much for joining. See you again next week, Tuesday. Thank you.